Okay, so this is the last part of the review, and by now we've done the first section, the second section, the third section, and so this is the last part. So this is the only part that you should have left to do. This first one, so this does not look like a plant because it's unicellular, but it does have chloroplasts in it. And as you know, anytime you see a picture that has the sun in it, it almost certainly has something to do with photosynthesis. So, let's turn this up like this. So when we're talking about photosynthesis, first off, what does chlorophyll do? Chlorophyll absorbs light energy, and that light energy drives the whole process of photosynthesis. Okay, so while we're at it, let's talk about what the equation for photosynthesis is. What do plants require? They require from the air carbon dioxide, which enters, diffuses into the cell. They also require water, which in typical plants is absorbed up through the roots, but again can absorb into the plant through diffusion. And they produce glucose, which is the key main purpose here is to actually make glucose, because glucose stores a great deal of energy, which plants and all organisms then use in the opposite direction to produce ATP in cell respiration of glucose. And the other product is oxygen. So these ones here, things you use that you start off with, are your reactants, carbon dioxide and water. The things that you produce, obviously your products, and in the case of photosynthesis, that's glucose and oxygen. Obviously you need to know the equation for photosynthesis. Okay, this one we're talking about blood types. We have two people, one who is heterozygous A, another who is heterozygous B. So just like any other genetic cross, what are the possible blood types? We're going to have to do a genetic cross. And remember, blood types, unlike most things, have non-Mendelian inheritance in that we have codominance. So I'm going to write geno and pheno. And again, anytime you get a genetic cross where you have to do a Punnett square to work the answer, that means you have to do a Punnett square. You can't get away without doing it. So looking here, we have an A and a B, and I know based on my rules that A and B are codominant. So my genotype is going to be AB. The phenotype is what blood type that would be. So that person would be blood type AB. Box number 2 is going to be A and an O. So they would be heterozygous, because they've got two different things. Because A is dominant and O is recessive, their blood type would be A. Look at this one. They're going to be heterozygous B. Because B is dominant and O is recessive, their blood type would be B. And the last one here, homozygous recessive O. So that's how you're going to get blood type O. Okay? Remember the key bit here, that A and B are codominant. Which means that they will cover up anything you have an O where it's recessive. Okay, this diagram here should be pretty familiar to most of you. This is the cell cycle, And remember, the cell cycle starts at the beginning of G1 and it goes this way round. So you have G1. What is this one? It is the S phase. Then G2, M, G, and cytokinesis. You start with one cell, and at the end of cytokinesis, you're going to end up with two identical cells. What happens during the S phase? This is the only question you're ever really going to get asked a lot about. And what gets made? The DNA gets copied. So you have DNA synthesis. Remember, it's semi-conservative DNA replication, which means the DNA opens up and makes two identical strands of DNA through complementary base pairing. Remember, up here, A with T, G with C, but the original strands are saved in the two new strands. But also, right after G1, right, and the S phase, and right before the M phase, mitosis, you get checkpoints. Okay, why are these checkpoints important? Well, for a bunch of reasons, but one, if your DNA is not copied properly, remember, changes in the DNA are mutations, and mutations typically are not a good thing. So you want to check the DNA is copied properly or accurately. That's the first thing. If it's not copied accurately, we go back into the S phase, they fix everything, and then proceed onwards. If it can't be fixed, the cell, remember, the cell self-destructs, and that's the end of that cell. You do not want mutated DNA being copied over and over again, because changes in your DNA and uncontrolled cell growth is what leads to tumors, and tumors end up as cancer. And then the other thing, so if your DNA is not copied, your cell's not going to be healthy. We want cells to be healthy and we want proper growth. Okay, proper growth. Remember, the cell cycle, that M stands for mitosis. Mitosis is when we're talking about cell growth. You will notice up here at the very beginning when we talked about meiosis, we only talked about how gametes are produced. Meiosis is not related to the healthy growth of the cell. Meiosis is related exclusively to the healthy production of gametes. So growth is not part of meiosis. Growth is part of the cell cycle. Right here, cell cycle. It's part of the cell cycle and mitosis. Remember the one where you start with one and you end with two. Okay, I talked to you a little bit about dihybrid crosses earlier, and I said this ratio here is the only ratio you really need to know when it comes to dihybrid crosses, 9:3 to 3:1. This is only about dihybrid crosses. And this is more a way for you to check things rather than to test, to take a shortcut. Because if you get that ratio, you better hope that the parents were heterozygous, big letter, little letter, for both characteristics. So you see, that would be heterozygous A and heterozygous B. And you better make sure that both parents are big A, big B, little b, whatever the letters are. You need a big letter and a little letter for both characteristics. In fact, that probably needs to fix that, like that. Big A, big B, little b. Two heterozygous genotypes. And just to reiterate the whole thing about codominance, okay? So this COD Okay, codominance, CODOMINCE. So in rabbits, fur color can exhibit codominance. So here is a white bunny. Here is a brown bunny. And if they do what bunnies do and have bunny babies, you can imagine and predict, because of codominance, that the baby bunnies, can't think what a baby bunny's called now. I think they're called kits. A baby bunny would have both color furs of both parents. So it would have brown and white. It might be patches. It might be that word roan, where they kind of is all equally mixed together. You can see the white hairs mixed with the brown hairs. But you're going to see in codominance, if capital W is for white and capital B is for brown, the genotype of the offspring would be, I guess it would be BW because we put them in capital in alphabetical order, but you would see both characteristics in the heterozygous phenotype. And that is part 4 of the review. If you've done this correctly now, it will look like this, kind of crazy. How you stick this in your notebook is absolutely up to you, but it does need to go in your notebook. If you want to do the fancy stuff, then you need to go check, I think you need to go check the first video on how to set up your review.